Well, hi, folks, and welcome back. Now we're going to talk a little bit about what happened after Mendel. The first person who comes to mind is Thomas Hunt Morgan. He basically rediscovered everything Mendel was working on. Remember, Mendel was contemporary with Darwin. He was working in the 1860s, 1870s. Thomas Hunt Morgan comes along in about 1933. Morgan started working with looking at these, what Mendel called particles, moving traits from parent to offspring. Well, Thomas Hunt Morgan coined the term gene, which we now use as in genetics. That is the particle, and I use air quotes when I say that, that Mendel was working with. He also coined the term chromosome, and we'll explain why that is in just a minute. When he brought his Mendel's work into play, he started looking at mutation and as well as inheritance, and really through all of these things was able to revolutionize modern medicine. Thomas Hunt Morgan did win the Nobel Prize, and he allowed us all to understand through his work. The first thing that we're looking at here, what is that? Looks like kind of worms with bands on them. Well, the hint here is what's in the background. That is this bluish, purplish dye. This is actually what this item is named after, because Thomas Hunt Morgan realized that if he pours dye onto the slide, it made things show up a lot easier to the eye. So this color, which is the key here, is what gave that thing, what we're looking at, this banded worm thing, its name, chromosome. These are chromosomes, human chromosomes. Chromo means color. And the color would be taken up into these bands, which are otherwise unseeable without this dye squirted all over the slide. So by counting these bands, we can see certain things. And now we know chromosomes are where we find a lot of different aspects of our genetics and health. This is before DNA, mind you. So what are chromosomes? Well, first and foremost, most of you probably know how many chromosomes we have. We've got 23 pairs of chromosomes, meaning 46 in total. You get 23 from mom and 23 from dad. Oh, these are the building blocks of life. And really, within these chromosomes is DNA. So let's look at what chromosomes gave us in Thomas Hunt Morgan's day. It gave us an understanding of where things that are abnormalities that are genetic will show up. For example, abnormalities in one of the 22 numbered pairs of chromosomes. Wait a minute, if there's 23 pairs, why are only 22 of them numbered? Because the last one is, anybody? If you had biology or anything else, you know those are the sex chromosomes, X and Y. The X chromosome, everybody has. XX are women. They are a homozygote, right? Sound familiar? XY, male, heterozygote. We all get X from mom because mom has nothing else to give away. She's a homozygote. We get either an X or a Y from dad. He's heterozygote because he's male. So those are the sex chromosomes, and things can go wrong in those as well. But there are a lot of chromosomal abnormalities that we know about, like Lou Gehrig's disease or Down syndrome, trisomy 21. Trisomy, meaning there's 3 chromosomes. Of which number? 21. You're supposed to have a pair, but if you have a third one, it causes issues that we see in Down syndrome. Other abnormalities can happen, like you can have an insufficiently formed one. Alzheimer's disease seems to show up at chromosome 14. This is not an exhaustive list of some of the abnormalities that we see associated with chromosomes, but it gives you an idea of how many there are. There are thousands of these, in fact. Thomas Hunt Morgan was also looking at mutations. And what he decided to look at instead of pea plants were fruit flies. Why fruit flies? Well, they have a very, very, very short lifespan. Furthermore, because of that, they have a very, very short gestational period and very short generation. Using the fruit flies, he could look at several generations within the matter of days, if not hours, with fruit flies. What he was looking at in this image is the different colored eyes that they may show up with. He also went on to look at other mutations, and he created a few new rules that we didn't quite fully grasp even after Darwin. And Darwin touched on this because he was so far ahead of his time, but most people didn't pay much attention to what Darwin was saying about mutation. For example, here we have a fruit fly with messed up wings. What's that tell you? Well, it's a fly that can't fly. That can't be good, right? So Thomas Hunt Morgan said, these mutations will either be deleterious, meaning bad, advantageous, I'm sure you know what that means, or neutral. And the vast majority of mutations are in fact neutral. Like for those eye colors, it doesn't hurt the fly in any way if its eyes are a different color. It doesn't seem to affect its mating chances, doesn't affect its ability to see, doesn't affect anything, apparently. Similar to human eye color. Blue, brown, green eyes, doesn't matter. Doesn't affect anything. Those are neutral mutations. But this is a fly that can't fly. You would assume that's a negative mutation, right? You would assume this is a deleterious mutation. Well, is it? How do we get this super close-up picture of the fly, and what's the fly doing? The fly is eating, even though it doesn't have working wings. It doesn't need them because it lives in a lab, and it's going to eat just fine. Now, this is our first clue into what is so important about what Darwin was saying. Remember how I said survival of the fittest is not a very accurate way of describing his natural selection hypothesis? Well, here we go. Why is this fly not affected by its messed up wings? Because of the environment it's in. Remember, this fly lives in a lab. It's being fed. It's probably sitting on a banana peel right now. So it's being fed just fine. Those messed up wings have nothing to do with its ability to survive, get enough to eat, or make a bunch of offspring. It won't hurt it in one way or the other because it's in a lab. We'll talk about how much environment means to mutation in a little bit. Here's another fly. Hey, this one's got 4 wings. That's got to be a good thing, right? We're Americans. Bigger, better, faster, more. Yay! More. Must fly better. Not necessarily. It may actually be that those second set of wings doesn't even work. Or if they do, they may actually get in the way. Or it may not help it fly at all. But again, this fly lives in the lab, so it doesn't matter. It's just something interesting. In fact, it may get more to eat and be helped along more because it lives in the lab and they want to study it. This brings us to what happened after Thomas Hunt Morgan. This is Watson and Crick. By the way, they're missing one integral part of their team, which is unfortunate. We'll talk about that in a minute. Watson and Crick are the guys who are associated most with, probably saying it right now at the screen, DNA. In fact, these guys also won the Nobel Prize for discovering the shape of DNA. We now know, and most of you are probably fairly familiar, that DNA comes in this shape. It's called the double helix because you guys can see there's a helicoil here and a second one here. And they spiral around a center. And in those centers, you see those little rungs of the ladder. Those are different protein pairs that make up the DNA code. This is what drives, ultimately, all the rest of the cells in the body to become what they become. So Watson and Crick discovered the shape of DNA. But did they really? First of all, let's look at what they were looking at. This is the image. This image was taken with an X-ray, and the reason for it is X-rays protrude out from there in a spherical way. In other words, they go out in all different directions, which means that if you put something very small up close to the emitter and put the photographic paper or the film far away from the emitter, it will make it bigger. So that's what happened here. And what you're looking at is a top-down view of the double helix. You can see this sort of circular pattern around the outside. Those are the helicoils, and in the middle, those are the rungs of the ladder that make up those protein pairs. But Watson and Crick did not make this photograph. That's the third person in their team. Rosalind Franklin. Rosalind Franklin was working with the X-rays. She came up with the idea of, hey, we've got something super tiny. I want to get a picture of it. I'm going to use X-rays. She did so. This is in the 1950s, 1955. She worked tirelessly to do that. She finally got a good image. She brought it to Watson and Crick. She said, this is what you're looking at. She told them the shape that they're looking at. They then went on and published a whole bunch about it. At first, they included Franklin in all the publications, but tragically, Rosalind Franklin died at a very young age. Working with X-rays, guess what she died from? She died of cancer. And even more tragically, she died of cancer before the Nobel Prize was issued to them. And if you're dead, they don't give you the Nobel Prize. It's not awarded posthumously. It's one of the rules. So Rosalind Franklin was not included in the Nobel Prize winning thing. And unfortunately, she's faded a little bit in our remembrance. So remember Rosalind Franklin when you're thinking of Watson, Crick, and Franklin. To this day, you can get bobbleheads of Watson and Crick, but there's no bobblehead of Franklin. It's unfortunate. I think she really was robbed of her notoriety that she deserves because of that. It's not, by the way, because the Nobel Prize laureates are sexist. In fact, one of the first Nobel Prize laureates was a woman, that is Marie Curie, who also died of cancer because also was working with radioactive things. It's crazy stuff.